On the 8th of March 2023, scientists in Western Australia were using a method called scraping to bring up insects from a drill hole. Somewhere among the media, they found two workers of a previously undescribed species of ant, which you see before you. It was a member of the genus Leptanilla. This species was named in their paper for the first time, Leptanilla Voldemort, for their resemblance to the character from the series Harry Potter. This genus was first described by Carlo Emery in 1870. The workers used to describe the first species of the genus were discovered under rocks in the mountains of Corsica by an associate of Dr. Emery's. Since then, this genus is usually found either by winged males which have flown from the nest, underneath large rocks or logs, as Emery's specimens were, or in the case of Leptanilla Voldemort, in a deep drill hole using special techniques. The genus has 67 described species, all come from the Old World and all share the same lifestyle. This lifestyle is described by the term hypogeic, which means that they are fully subterranean and do not even come up to feed. The only exception is the winged males who leave in search of another nest to mate with a female there. This life history is the reason for their morphology. Workers are extremely small, pale, and lack eyes. They have no eyes at all. Over time, they have lost them from disuse. While most have fairly elongated bodies, Leptanilla Voldemort are especially gracile. Every part of their body is elongated to such a degree that while mounting the specimens, both were damaged. The postpedial and gaster fell off the rest of the ant. In the paper, it is speculated that once again this morphology is due to their life history traits. They note that the other species of Leptanilla in the same region, Leptanilla swani, was first found underneath a large rock, while the new species, Leptanilla Voldemort, has only been found via subterranean scraping. This implies that Leptanilla Voldemort lives deeper underground than the other species of Leptanilla, which possibly explains the unique morphology to some degree. The only other fairly gracile species of Leptanilla seems to be Leptanilla leventa, which is found in hollow subterranean pockets in Iran, which makes it possible that this is a similar habitat to where they were found in Australia. While the unique biology of Leptanilla Voldemort is unknown, other Leptanilla species exhibit some interesting feeding strategies. The first interesting feeding strategy is found in a paper published in 1990 about the biology of Leptanilla japonica, where it was observed that workers refused most every prey item except for geophilomorph centipedes. The following is an account by K. Masuko of how a colony feeding occurred. At 13:20 on the 23rd of March, 1982, an intact geophilomorph centipede was admitted to the nest. At 13:34, a worker first succeeded in stinging the prey, which it had seized with its mandibles. At 13:54, 12 workers were counted attacking the centipede at a point directly 7.5 centimeters from the brood pile. The prey was almost completely paralyzed. At 14:07, about 30 workers were present around the victim, which had not moved since 13:54. A few workers were then first observed to drag the prey towards the brood pile, though most others simply chewed and licked its surface. At 14:43, the centipede had been dragged to a point 6 centimeters from the brood pile. At this point, the first larval transport towards the prey by a worker occurred, but the transporting worker turned back with the larva to the brood pile about halfway to the prey. At 14:48, the queen first departed the brood pile and moved all the way to the prey, but soon returned to the brood pile. After that, she repeated similar lone shuttles between the brood pile and the prey site 3 times. At 14:56, a worker carrying a larva first arrived at the prey, now 4 centimeters from the brood pile, but did not settle there. Between 14:59 and 15:01, workers carried larvae one after another along a trail between the brood pile and the prey site, with the result being that all larvae were ultimately transferred. On the following day at 10:45, the prey body was found largely consumed, with most of the larvae attached to it. It was further written that it was found at times prey would be moved to the colony, and at other times the colony moved itself directly to the prey. In a paper published the year before by the same individual, K. Masuko, it was found that the primary way the queen receives nutrients and how some of the workers feed in addition to prey was something termed larval hemolymph feeding. This basically means insect vampirism. The hemolymph is the circulatory fluid in insects, functioning basically as blood. While other ants are famous or infamous for doing this, Leptanilla japonica larvae were observed to have something unique, termed by the paper as a larval hemolymph tap. This was basically a duct from which hemolymph or some other nutritious fluid was excreted for adult consumption. As I said before, it seems the queen got almost all of her nutrients via this process, and later became physiogastric. At the same period she fed most often, which means that she would fill up with many eggs, before being observed to lay many of them during the same period. Masuko observed 50 being laid in a period of 3 days. It is also noted by the paper that while other Dracula ant queens were found to try to suppress workers from drinking hemolymph, in Leptanilla japonica, workers were freely performing this action even in close proximity to the queen. Whether or not Leptanilla Voldemort also feeds this way is unknown, but likely. While not overly important, the method by which this new species was discovered and how similar methods have discovered various other species is highly interesting. The method used to find Leptanilla Voldemort is referred to as subterranean scraping. This method is essentially where a drill hole, 25 meters deep in this case of the discovery of Leptanilla Voldemort, has a net with a capsule placed down it and repeatedly pulled up, scraping the sides and gathering any media into it. The media is then preserved with alcohol and sifted through afterwards. This method has its limitations mainly in that any media could have come from any layer of unknown depth. Beyond this, other methods are mainly subterranean baiting and soil sampling. In the former, some sort of device with sugar or protein bait is placed at some depth and retrieved some period later, and its contents examined. Soil sampling involves taking large samples of soil and carefully sifting them for anything of interest. Both of these have their limitations, like scraping, however, but none will ever be perfect. While at first thought subterranean environments do not seem important, I would argue that they are probably of unknown importance and therefore of utmost importance until we investigate them further. Leptanillidae as a phyletic group may be a sister group to all other ants, which means that they may be of great importance to establishing the proper phylogenetic tree of ants, and every discovery should help. Many other barely studied insects are routinely found in studies on subterranean life. I am sure we will all watch this field of study with great interest from now on. All of my references can be found in the description.